So, as Phil said, and as Dan also talked about, I've been working with Dan this winter to explore traits data that may enhance restoration applications, and particularly in regional databases. And we were really curious about the Consortium of Pacific Northwest herbaria, which we got to from curiosity about University of Montana's herbaria. And if you don't know what an herbarium is, it's where botanists bring their collections for identification and preservation. So the University of Montana has an herbarium that has collections from all over western Montana and beyond that have been acquired over the last 200 years. And there's a huge amount of data with the consortium, which is, it says 57, but there's actually 28 herbaria that you can reference on the website, and then 57 smaller herbaria that come from there. So there's one down in the Bitterroot Valley, which is a smaller one that would have its collections go to University of Montana and then connect to the database. So as it says here, there's 3.6 million specimen records, and we wanted to see if we could get any information from that that would help us in selecting plant materials. So one of our main questions was, can we use the herbaria databases to inform restoration? And we're going to go check out that website right now. And I'll show you just how to do a little search. Actually, I don't want to go there yet. So the strength of the database are that it has a huge amount of data, and it's just plain cool to be able to go through all that. It provides historical records dating back to Lewis and Clark, and also has the renowned botanists for this region putting collections into it. One of those botanists that helped us a lot going through the herbarium site was C. Leo Hitchcock, who wrote the Flora of the Pacific Northwest, and he actually worked at the University of Montana in the 40s for a few years, and many of his collections had really great habitat descriptions, which was helpful. So there's accessions from 28 herbaria. It's geographic location for many accessions and spatial data for that, which is hugely helpful. There's polygon search feature, and there's a mobile app that has potential for field work. So some limitations, and there are certainly a few, is that it only shows the presence of a species. Often, although there is spatial data, it's inaccurate because it's just before the revolution of GPS, there was just a county, or it was by this road that may not even exist anymore. It's labor intensive to filter for habitat, and in some cases, it's a rare, unique collection for that site. So, for example, one of the greatest limitations of it is that you can't search for a habitat, and it's only limited by locality. So this is the only way to search for a habitat on the site. And so if we put in grassland in the state of Montana, it brings you back 28 matching records, which is actually less than I've gotten in the past searching on this, so I'm not sure what I did differently this time. But usually it brings back 77, and we know there's more than that of grassland species in Montana. So that was the most labor intensive, is sorting through that. And one way that you can sort that is an asset on the herbarium site is the so the 77 matching records found, 53 displayed on the map, is the polygon feature, which I'll review in a second. So one way that we could apply this, so the question of can we use the herbarium database to inform restoration is still unclear. And a few questions that we wanted to use to flesh that out was, if we could figure out what ranch populations or introduced native species are genetically isolated, and do historic and or regional records suggest that there's genetic connectivity. So there's a few factors that that influence gene flow and relative isolation at the ranch. The position of the MPG in the Bitterroot Valley. There's physical barriers to gene flow. There's the rain shadow of Lolo and the Bitterroot. And there's anthropogenic disturbance that are inhibiting gene flow or allowing it in certain cases. So we specifically searched for grassland species for this pilot exploration of the herbarium database. And our grassland communities need to connect to other grasslands through grassland corridors. And we were hoping that the herbarium accessions would give us a historic image of connectivity. So the gene flow has to occur along the bottom of valleys for grassland species. And in order to do that, it has to snake through the valleys and go past the physical barriers, like the Bitterroot, the sapphires, and the rattlesnake and northern Bitterroot. So one of the most promising corridors is coming through Hellgate Canyon. But then there's also coming from the north, the Flathead grasslands, and also from the south, although that is questionable, of coming from the Big Hole or down in Idaho. So this is a few of the grassland prairie ecological systems that connect to western Montana. And of course, we're in the northern Rocky Mountain, lower Montane foothill, and valley grassland. So another physical barrier, well, a climatic barrier to gene flow is the rain shadow that we're in. So this figure shows you the average annual precipitation in the western United States, unfortunately only up to 1990. I got it from the University of Wyoming. But you can see zoomed in there is the Bitterroot, and that's where all that rain is getting squeezed out, and we have that very arid rain shadow effect at the ranch. Bootalua gracilis is a species that demonstrates this genetic isolation at the ranch. This is came from the website and is the collections that we've had on or the observations we've had on the ranch. The ones down in the north center and pivot were planted, but all the rest were found here. They're remnant populations, and it's a surprising find of Bootalua gracilis on the ranch, and it's because of its unique position. The ranch's unique position is what is a thought about that. So as you can see, according to the herbarium accessions, we're on the western edge of Bootalua gracilis's range, and there's not much between us and Butte, or between us and Helena. So that's a demonstration of its isolation, and it's something we'd like to explore further, maybe. And this is the historic collections of Bootalua gracilis, the earliest being in 1923, and the most recent being in 1993 in the Woodchuck Creek drainage just near the ranch. So another species that we don't have remnant populations on the ranch of, and we don't have a record of on the ranch, is prairie coneflower, but Dan was definitely curious about why that was, because we have, because it seems like something that should grow here. And this is a picture of it in the north center pivot in 2012. It's been planted on the ranch, and we are currently planting it on the ranch. And it has a historic range south of the ranch and north of the ranch are collections. So that's a good a good indicator that supports our use of it on the ranch. And here's the historic collections, the earliest one being in 1909, and the most recent in 1998. So this is a map of the accessions of prairie coneflower from the Pacific Northwest Herbaria database. And opposed to Bootalua gracilis, or very similar to Bootalua gracilis, demonstrating its genetic isolation. So another question that we were using to guide us in figuring out if this would be a useful tool was, what plants have historic presence in the region and do not appear on current ranch species lists? And we chose a 4-county area. They're Valley, Missoula, Granite, and Lake counties, and we chose that range because of the genetic connectivity to the Bitterroot Valley. They all have potential corridors. So if you go to the Pacific Northwest Herbaria Database search site, you can search for multiple herbaria or just a single herbarium. And if your search isn't confined enough, then you won't be able to search multiple herbaria because it can only bring back so many queries. But this is where that polygon feature came in, was a good asset for this exploration, was that you can, instead of, to work over that limitation of not being able to search by habitat, we drew a polygon around the valley regions in order to eliminate high elevation species and forested species. Of course, we also got all the habitats that are in the valley besides grassland, which is where the labor, where it's not a very realistic tool because it is very labor intensive to sort out those habitats. And this is what we came up with for Valley County collections. So that's 2,952 matching results for all of the species that are at a lower elevation in Ravalli County. Then you can download it as a text file and put it into an Excel database, and we sorted out species already present on MPG's plant list, sensitive and rare species, as well as non-native species, and we filtered for grassland species. And as I mentioned, it's a rough polygon of grassland habitats within each county. Its geographic information is often inaccurate. Sometimes those points would be in the center of the valley, but they had been collected at the top of the Bitterroot Mountains upon further investigation. And it's labor intensive to filter for habitat. So after that search of the 4 counties, we came up with a 39 species list of potential grassland species that we could apply to restoration treatments on MPG. One of those is Pescopyrum smithii, western wheatgrass. Dan had intended to use this on the ranch and was surprised not to find it on the species list. And our search on the herbarium database supported that because it's found in Ravalli County and Missoula County, has a historic record, and we're right in the middle of its range. So it has just been applied by the field crew onto some of those lighter soils that were moved during the flood last month. So we'll see how that goes. And some future applications for the herbarium data. Did we answer the question of if we can use it or not to find restoration materials? Some ways that we could is to further investigate historic presence of genetically isolated species found on the ranch. And it's also a great tool for if there is a certain species that we have good access to for seed supply and we don't have on the ranch list and we don't have the background to know where, if it was in Missoula or Ravalli counties, is to search on the herbarium site. So another step would be to extract promising species from that list of 39 that maintain healthy ranch populations with adequate genetic connectivity to off-ranch populations and contribute to the function of ranch habitats. And if we do find species from that have potential, we could use the geographic information from the herbarium site to find populations from which to collect seed. I've also been working on a few other databases. I've also been going through a few other databases for plant trait information this winter. One is the Plant Trait Database, which is actually an international effort. And it's just pooling plant traits information from wherever it can get it. So we could share our data with it and also receive data from the TRI database. and also the USDA plants database characteristics. We spent some time this winter sorting through that and trying to find out if we could apply characteristics and functional traits gleaned from that information to restoration practices. And this is my references. And I wanted to thank Marilyn Marler for, at first we went to the UM Herbarium site, which is from the early 2000s. And we're trying to go through there, and then I called Marilyn and she directed me to the Consortium Pacific Northwest Herbaria. And also the botany crew for going through those lists and helping with their knowledge.